My name is Chris Somerville. I'm a scientist in the Carnegie Institution and a professor at Stanford University and a scientist at Lawrence Berkeley National Laboratory. In this segment, I'm going to be talking about the technical aspects of making cellulosic biofuels. To begin with, I want to just address what's meant by biofuels. And in general terms, there are four ways, or there are currently four types of biofuels. First one can, of course, just burn plant biomass to produce heat, and the heat can be used for various purposes. Diesel can be made by plants, and I'll illustrate that in a moment. Sugars can be converted to ethanol by fermentation, and cellulose can be converted to ethanol or other fuels by fermentation. So I'm going to start off by just briefly describing some of the issues associated with these, and then I'm going to focus mostly on cellulosic biofuels because that's where the really large-scale opportunity seems to be. If we look in this slide, what one sees is a section of a single cell. So you can see over here, here's a piece of the cell wall, and these white bodies are fat globules inside the cell, and the cell is from an embryo in a plant seed. So many plants store large amounts of oil as carbon reserves for embryo development during germination. So these oils can be produced in relatively large amounts and in fact are responsible for about 30% of the calories in the human diet. In plants, the principal component of the oils is triglyceride, so a molecule in which the glyceride backbone or glycerol backbone is esterified with 3 long-chain fatty acids. And these fatty acids can be cleaved off and converted to methyl esters with very high efficiency. It's something that can actually be done on your kitchen counter with reagents you can buy in a grocery store. So for that reason, there's a lot of interest in making so-called biodiesel by reclaiming used cooking vegetable oils, for example, or tallow from animal processing. And indeed, there's currently about 65 biodiesel plants in the United States and many more elsewhere in the world, and there are about 50 more currently in development. And it's a concept that's much in the news. And indeed, it's a good, if the vegetable oil or the tallow is available, it is a good technology. The diesel engine was in fact invented when it was first invented. The fuel was vegetable oil, so it's very compatible with the current engine design. However, there's if one is looking at the big picture in terms of to what degree biodiesel can contribute to our energy needs, it's not very promising. This slide is from the Congressional Research Office, and it's showing basically that we use about 140 million gallons of gasoline in the United States each year. We use about 60 billion gallons of diesel. And according at least to the Congressional Research Office, if we converted all the plant and animal fat in the United States to biodiesel, we could only get about 3.5 billion gallons. Of course, that would remove a large amount of the calories from the human food supply. So that's probably not really feasible. And in fact, the 65 plants that I referred to are shown over here. And as you can see, they don't really even come above the line on the national needs. So there's probably not a very good opportunity in biodiesel. One of the reasons is that the main oilseed crops, for example, soybean in the United States is the big oilseed crop. It will only produce about 70 gallons per acre of oil. Now, by contrast, in the previous segment, I talked about cellulosic biofuel plants, or cellulosic plants that can make biofuels. And on an acre of a really productive grass, such as Miscanthus, we could probably get about 2,500 gallons of ethanol. So because land use is fungible, it seems very unlikely that in the future we'll be satisfied making 60 gallons of biodiesel when we could be making 2,500 gallons of ethanol. Now, there is one plant that does have much higher productivity, and that's oil palm, which is a tropical plant that grows within 15 degrees of the equator. And This plant, because in those regions the growing season is all year long, and the plants are very long, have a long lifespan. These plants are very efficient accumulators of oil. You can see that up in the crown of the plant they have a fruit that's a lot like an avocado, and as with an avocado, the mesocarp tissue contains about 35% oil, and the kernel, this white region in the center, is also contains a lot of oil. So these plants can produce up to 10 metric tons of oil per hectare per year, and in fact, a very large amount of palm oil is used worldwide. Of course, the problem with the oil palm is that in those regions of the world where it can work, there are a lot of tropical forests that are under pressure, and it needs to be examined the degree to which oil palm acreage could be expanded without seriously impairing biodiversity. So well, anyway, that's the story about palm. Now, in the context of biodiesel, another idea or another opportunity to produce biodiesel in theory is that many algae, marine algae, produce fairly large amounts of lipid within their cells as lipid droplets. And so there's interest in the possibility of large-scale cultivation of algae for that purpose. And indeed, a number of companies have sprung up during the past several years that are cultivating or trying to establish conditions for large-scale cultivation. This slide shows a reactor on the roof of a building in Massachusetts. I think this is a green fuels reactor, as it's called, the name of the company. And in this case, what they're doing is, you may just be able to see that these tubes are transporting our algae around, and then there's a kind of a big place where sunlight is incident on the algae, and the algae are constantly swirling through. And indeed, and there's a smokestack in the background in which gases some CO2 enriched gases are being bubbled through the cultures to stimulate photosynthesis. Now, although this looks promising, the challenge is the following with algae, and that is that sunlight contains about, on average worldwide, about 200 watts per meter squared. And since photosynthesis by algae is only at maximal about 9% efficient, it means that actually per meter of algal culture, the energy that's being captured on maximum efficiency is below 20 watts per square meter. And it can be very challenging to build devices such as this or many of the other devices that are currently under development when the return is only about 20 watts per square meter. To put that in perspective, electricity currently sells for as little as 3 cents per 1,000 watts or 1,000 watt hours. And so that gives you some sense of what the current challenge is. It's been the idea of actually releasing algae into large open ponds is also discussed, but there seems to be a biological problem there, because if one tries to grow large amounts of algae and applies sort of pressure on the population by harvesting it repetitively, that leads to selection for strains that can propagate rapidly, and that in turn leads to strains that don't produce any oil. So right now that's the kind of problem with generating large-scale algal culture, although it may be possible to solve it with some innovative designs in the future. When one talks of biofuels today, in general, it means ethanol produced from one of two main sources in regions such as Brazil. Ethanol can be produced from sugar, which is from sugarcane. Sugarcane is a very highly productive plant, and the canes can be harvested and crushed to release the sugar, and the sugar can be fermented with very high efficiency. So indeed, Brazil currently gets about 40% of its transportation fuels by fermentation of sugarcane, and that's done on only about 1% of the land acreage of Brazil. So for Brazil, that's been a very important way of establishing energy security and stimulating the indigenous industry. I think it supports about 1 million jobs in Brazil. In the United States, during the past several years, we've seen a rapid expansion of production of ethanol from corn grain. And this map of the US from the National Corn Growers Association shows the location of about more than 100 plants. You can see all the little dots scattered around the Midwest in particular are the sites where corn grain ethanol plants either exist or are being built. And in general terms, these plants are mostly owned by farmers or farmer co-ops, and so farmers are gathering together to process their grain into ethanol, and these have proven to be very profitable because corn prices have been low for the last 25 or 30 years. The reason that corn is an attractive crop is that it's quite productive, and the grain is mostly starch. So you can see over here, this is a section through a corn grain, and sort of everything above the embryo is starch. And starch is just a polymer of glucose, which can be fermented by yeasts with very high efficiency to make ethanol. The remaining part of the corn embryo, by the way, is fed to cattle as cattle food. So it's all used, but only the starch is currently used for ethanol production. In general terms, making ethanol from corn is, while it's proven to be very profitable for farmers, is not considered very desirable from an environmental standpoint because corn uses quite a lot of energy during the growth and harvesting and the land manipulations, so that typically it's thought that if one does a full life cycle analysis of how much energy goes into growing, harvesting, and processing corn, and compares it to how much energy is returned from the ethanol produced, it's currently estimated that there's only about 1.3 times as much energy recovered from the corn as goes into growing it. So from an environmental standpoint, that doesn't seem very optimal. And there are some negative environmental effects with the large-scale monocultures of corn, such as quite a lot of runoff of fertilizer, for example. So recent sort of predictions of where the ethanol industry in the United States or in North America is likely to go during the coming decades is shown here. And in general terms, it's thought that corn grain will probably, or corn grain ethanol will probably saturate at about 12 billion gallons a year. We're currently making about 6 billion gallons. And the reason for that is that, of course, until the ethanol boom began, corn grain was used to feed cattle mostly. And so that need has not gone away. So as the use for making fuel has increased, then the price of producing animals has gone up, and a new equilibrium will be established in which it won't become economically feasible to use corn grain for fuel. In that respect, I think it's worth noting that we've just witnessed, or we're witnessing, a historic, what I can think of as a historic event in which the value of a bushel of grain and a barrel of oil have come into some sort of an equilibrium based on the BTU content of those two, that is the intrinsic energy content of those two commodities. And so even though it's true that the boom in ethanol from corn and sugarcane during the last several years has pushed up the price of food, that will not continue indefinitely. I think from about here on it should track the price of oil. So because until large-scale production of corn ethanol started, there was the grain and energy were in different economic sectors, but now they're conjoined, and so a new equilibrium should be reached approximately now. All right, so in thinking about where this is going, since we can't meet our energy needs with corn grain, it's generally thought that what will happen is that during the coming years we'll start to see an increase in the production of ethanol from cellulose, or lignocellulose as it's sometimes called. And so mostly what I want to address now is what does that mean in a practical sense, and what are the challenges with doing that? Before I do, I want to address one of the general criticisms that is often voiced about making biofuels, and that is authors such as Pimentel and Pectic have argued that it's generally summarized in the press as that the amount of energy we get out of biofuels is less than the amount that goes in. And indeed, that's probably not true, but I want everyone to understand what their argument really is, because it has two components. And so even though they're critical of the technology, Pimentel and Pectic would agree that from their paper that if you just grow and harvest and burn a plant such as switchgrass, a perennial grass that's of interest for biofuels, you can get about a 15-fold energy return. So there's not really any argument whether plants can be grown as net energy sources. The real argument is the degree to which, when one converts the cellulosic biomass to liquid fuels, you lose a lot of that energy content. And so in their analysis, Pimentel and Pectic sort of look at the current process, and they add up all the energy costs that go into actually processing the biomass. And as you can see from this pie chart, which I constructed from their paper, they project that a lot of steam and electricity goes into making the biofuels, which is true. And so it certainly is, although there's a lot of disagreement about their numbers, it's generally true that the current processing of biomass to fuels requires a lot of input energy. And so people that are interested in making biofuels a possibility are really focused on this general issue. How can we decrease the amount of energy that goes into converting cellulosic biomass to fuels? And that's mostly what I'm going to address. I think that we can make a lot of improvements. So to understand that in general terms, we need to sort of look at the overall process of making cellulosic ethanol, and shown here. So over on this side, we start with a bale, it's ground up, and then typically the biomass fragments are then boiled in hot acid. And I'll show you in a few moments what that accomplishes. But in general terms, it breaks down the biomass into, it partially breaks it down into free sugars and fragments of biomass. The free sugars can then be neutralized and fermented to produce a certain amount of ethanol. The remaining solids are then usually treated with enzymes such as cellulases that break down the remaining polysaccharides, and then those sugars are neutralized and fermented so that at the end one ends up with ethanol. And the ethanol is then frequently distilled. So this is a pretty well-known process that emerged from 5,000 years of probably or more of making wine and beer. Now, to understand what the technical issues are in carrying out that process, let's start at the most basic level. What is plant biomass made of? And you can see that if we take a typical grass composition, about 40% of it is cellulose. Then there's a large component of something called hemicellulose, which I'll introduce in a moment. Another major component is lignin. And then everything else, basically the protein and the mineral nutrients, are actually quite a small proportion of the total biomass. So this slide is a blown up image of a piece of a pine board. And what we're seeing here, for example, there's a cell, and where my finger is, that's a cell wall that's surrounding the pine cell. And if we look up close at what that cell wall is, in this image, you can see, this is an electron micrograph, by the way, in which we're looking down on the surface now of a plant cell wall. And what one sees here is a highly interdigitated network of fibers that have a dimension of about 3 to 5 nanometers. And I'll just tell you sort of in advance that if those fibers are mostly cellulose that we can see, because although there are other polymers in the wall, they're a bit small to see. So cellulose is a composite material. The fibers, the 3 to 5 nanometer in diameter fibers, are made by hydrogen bonding approximately 36 chains of beta-1,4 linked glucose. So just to remind you, starch is alpha-1,4 linked glucose. Cellulose is beta-1,4 linked glucose. And so although they have the same sugar, it turns out one is easy to break down. The alpha linkage is easy to break down. We have enzymes, alpha amylases, that will degrade it. But the beta linkage turns out to be more challenging. The cellulose is actually made in the plasma membrane. You can see here there's a complex. And actually this is what my laboratory mostly is focused on, the research I work on specifically, is we're interested in how it is that the cellulose fibrils are made. And so what we've learned during the past few years is there's a large, probably 3 million Dalton complex sitting in the plasma membrane with about 36 active sites. And those active sites spin out these glucan chains which hydrogen bond to form the cellulose microfibrils. The hydrogen bonding of these microfibrils is one of the reasons that the cellulose is a very hard and tough material. It's not well hydrated. In addition, these fibrils are very big. So work from our lab has shown that the complexes actually can make about 350 nanometers a minute of these fibrils. And they make chains that are in full length, about up to 12 microns in length. So I guess that's about 14 or 15,000 individual sugars per chain. So they're pretty big molecules as these things go. Okay, well here's a kind of a very oversimplified image of what the cell wall actually looks like in my favorite model by Maureen McCann and Keith Roberts. What this is showing is the cellulose fibrils, the green, big green things, and they in turn are kind of coated with another material, these little red fibrils. Those are single molecules of hemicellulose. And hemicellulose, the most abundant one, is actually a xylan. What that means is it's a beta-1 4-linked xylose polymer. And each of those molecules is about 1,000 sugars long, to give you some sense of the dimension. And they hydrogen bond to the surface of the cellulose fibrils, as you can see. And then they may hydrogen bond, in fact, to several cellulose microfibrils. Additionally, there can be, depending on the type of cell wall, there can be other polymers in the matrix. So in this image, there are some sort of filmy-looking materials called pectins. And these are charged polysaccharides made of uronic acids mostly. Additionally, in certain types of cell walls, and mostly the cell walls that are of interest for making biofuels, such as the stalks of grasses and wood, about a third or a quarter to a third of the material is a polyphenolic called lignin, shown here. This is a kind of a typical example, and if you stare at it, what's striking about it is that there are a large number of linkages. It's very complex, and in fact, and it's very durable. So some of you may have heard of lignite coal. Well, lignite and lignin have the same root, and indeed much of the soil is also highly enriched in lignin, because the large number of linkages and the types of linkages, typically ether or carbon-carbon linkages, are quite hard to degrade. So it's a very durable material, and its role in the plant is probably to provide compressive resistance. So on this slide, to give you a sense of how this all assembles now at the nanoscale, I made a kind of a variant of the nice drawing by McCann and Roberts, and then amplified a small region of it to give you a sense of the high resolution, how these things all fit together. So maybe you can just barely see there's the green cellulose microfibril, and then it's covered with the hemicellulose strands, and then the lignin is in some way covering or wrapped into this material. And in fact, we know that the lignin is actually cross-linked to the hemicellulose, typically through so-called ferulate esters. So this is a very difficult material to really understand. The size of most of the components is below our ability to image with current imaging technologies, and we don't really know what the fine scale structure of this is. And in fact, that's one of the big research objectives that people in the field have right now, is trying to see what the nanoscale structure is, so that we can try to envision ways of breaking it down. For example, if it has this kind of structure shown here, one explanation for why the biomass is so hard to degrade is that perhaps the lignin occludes the biomass, the polysaccharides, so that enzymes can't get at it. And indeed, that would be compatible with evidence such as shown on this slide. So what this slide is showing is that the rate of hydrolysis, so on that axis is hydrolysis, this axis is on the bottom is the amount of lignin in the biomass. And you can see that there's an inverse relationship between the amount of lignin and the rate of enzyme-mediated hydrolysis. And we think that's due to the fact that because the lignin is kind of coating the cellulose and hemicellulose, the enzymes just can't get at it. So one of the things then that the pretreatment that I mentioned earlier, the boiling in hot acid or related kinds of treatments, what we think that does is that it first of all, it disrupts hydrogen bonding, causes things to loosen up, and secondly, it causes sort of non-specific hydrolysis of the hemicellulose. So the hemicellulose sugars start diffusing away, and the linkages to lignin start getting broken. So everything starts to loosen up, and the enzymes can penetrate. So then typically when, or in the past at least, until we were a bit further along in our thinking, the original approach to that idea was to say, well, let's get rid of the lignin. So actually a great deal of research has gone into understanding how lignin is made. It's shown, the pathway is shown on this slide. So up in this corner you can see that lignin starts from phenylalanine, and then through a series of transformations one ends up with a series of so-called phenylpropanoids. So phenyl groups, hydroxymethyl or hydroxylated phenyl groups with propane side chains. These are secreted out into the cell wall, and then they're polymerized in a free radical polymerization reaction. And in fact, that's why so many different linkages exist in lignin, is that there's about 4 major phenylpropanoids, and then they get secreted into the wall where they polymerize in all possible combinations. It's one of the few processes that I know of in which non-enzymatic polymerization takes place on a very large scale. Now, many years ago, I and other people made mutants of higher plants that were defective in lignin, and one of them is shown here. These represent 2 sections through stems of Arabidopsis. And what one sees here in those cells, those are the xylem cells, and the blue color reflects actually lignification of the cells. So these are cells that transmit water. And down here you can see, actually right there, you see there's some cells that are kind of collapsed. And so those are the cells in a mutant, a type of mutant at least, that is defective in lignifying the secondary cell wall. And so what happens is, if the lignin is not present, the walls don't have enough compressive resistance, and they collapse under the negative pressure of transpiration. That is, as the water moves through the vascular tissue and out the top of the plant, a negative gradient is formed, pressure gradient in the tissues. And so this in turn causes the plants to have growth defects. So unfortunately, it doesn't seem very likely that we can simply solve the recalcitrance problem, as it's called, by significantly eliminating lignin. There are many conceivable alternative approaches, but one approach that we're exploring, that the field is exploring right now, is to make a different type of lignin. And to explain that, our basic idea is that because lignin is made in a free radical process, it's possible that if we made a kind of a molecule or engineered plants to make a type of molecule that could be incorporated into lignin, but that had a cleavable linkage, we might get lignin that still has the right physical properties, but that could be degraded more easily. To give you a sense of that, this molecule, pinoresinol, is a normal component of lignin, and we can show, or it has been shown by other laboratories, that if one feeds this molecule to a plant cell, it gets incorporated into the lignin, as expected. So we think there's a possibility that we could develop plants that produce other types of related types of molecules, such as this molecule here called rosmarinic acid. It's made by many plants, but it's not secreted into the cell wall. And the nice things about rosmarinic acid is it looks a lot like things like pinoresinol, in that it's got a pair of phenyl groups with hydroxyls, but it has an ester linkage, and that ester linkage is readily cleavable, both enzymatically or by simple chemical treatments. And so one possibility is that we may be able to engineer plants to make and secrete rosmarinic acid, and then we get a lignin that's got good properties, but we can remove it under much more benign circumstances. Another general problem in the field is that once lignin and hemicellulose is removed by the hot acid treatment. So I just want to make it clear, in case I haven't up to this point, one of our goals in making modified lignins would be to get rid of the hot acid treatment. The hot acid treatment is one of the things that imposes a lot of costs on the overall process. That is, you have to heat it up to a rather high temperature, and you have to engineer containers, you might say, that can withstand highly corrosive treatments, and so that imposes a lot of cost on the overall process. So we would like to move towards a system that's basically physiological, and the reason we think it may be possible to move towards a much more physiological circumstance is that, first of all, many organisms, such as cattle and termites, are able to degrade lignocellulose, and compost heaps do it, and forest floors. So there's lots of reason to believe that that since nature can do it, that we can find a more benign route. Now, a component of doing that is going to be to find more effective enzymes. So under the current process, assuming we could deal with the lignin problem by some other method, we're still going to have the problem of breaking down the cellulose. And there's a class of enzymes called cellulases that can do this, but they have very low turnover numbers, and that results in a very large amount of enzyme being used. So 10 years ago, just the cost of the enzyme for making a gallon of cellulosic ethanol was about $5 just for the enzyme. During the past 10 years, that's been brought down to about 50 cents a gallon for the enzyme, but that's still quite a lot of enzyme. To put it in perspective, the starch-based starch-based ethanol production requires about 2 cents worth of enzyme. So this rather fanciful slide shows an artist's rendition of a cellulase sort of abstracting a molecule of glucan from a piece of cellulose and hydrolyzing it. In fact, of course, this can never happen. The cellulose can't be sort of pulled out of or the glucan can't be pulled out of the cellulose chain, and that's part of the problem. In terms of finding the optimal enzymes, recent advances in high throughput sequencing have created entirely new possibilities. So for example, these days it's possible to sequence a whole microbial genome in a morning and to annotate it with quite high efficiency at relatively low cost. So experiments are underway to understand, for example, how termites are able to chew wood and extract sugars and nutrients from them. It turns out it's been known for many years that the termites have about 100 species of bacteria in their guts, and now these are being systematically explored to find out what it is, what kinds of enzymes those bacteria produce and secrete, presumably into the termite gut. Similarly, ruminants such as cattle also have about 100 species of bacteria that work as a consortium to break down hay in the rumen. And those are being sequenced, and the enzymes will be characterized during the coming years. Compost heaps and forest floors carry out the same process and are similarly being explored by metagenomic approaches. In some cases, the organisms can't be sequenced, or they can't be cultivated, so the whole mixture of microbial biomass the DNA is extracted and is being sequenced, and then with a certain probability, the genomes can be reassembled from the total sequence. Additionally, during the past decade or so, there have been tremendous advances in structural biology and protein evolution methods that have raised the possibility that we may be able to improve the catalytic activity of cellulytic enzymes by rational methods or by selecting for improved enzymes in high throughput screens. And finally, I think there's a distinct possibility that we can synthesize novel non-enzymatic catalysts that might be useful. most of the organic chemistry that's done on an industrial scale in the world is actually done with synthetic catalysts rather than with biological catalysts. And so that's not really been explored yet in the context of biofuels. So I think there's a very interesting possibility to see whether new organometallic catalysts can be developed that are able to hydrolyze glycosidic linkages with a high efficiency. So I see a lot of opportunities that really build on modern advances in biology and chemistry. Finally, I think a very interesting area that illustrates how little we know in some respect about what the possibilities might be is kind of illustrated in this slide of a drawing of what's called a cellulosome. And the importance of this is that, or what the authors are trying to illustrate with this slide is that many microorganisms don't simply secrete enzymes into the milieu around them where they have some activity, but they actually secrete a complex that they then attach. As you can see over here, this complex is attached through this scaffold and to the surface of the microbial cell, and then a series of enzymes act in some synergistic way that's poorly understood to accelerate the hydrolysis of cellulosic biomass. It's not really known how these work, and in fact it's not really known yet what the structures are or even what all the enzyme activities that are associated with them are. But it's an active area of research that I think illustrates one of the many opportunities that exist to use modern biology to discover new principles of how biological systems are carrying this out. Now, another important component of increasing the overall efficiency of the process is that, as I've noted earlier, biomass is not just composed of cellulose or glucose, but in fact there's other polysaccharides containing other sugars such as xylose. And to make an efficient process, all the sugars, in fact, have to be converted to fuels. So at present, although we know many microorganisms that can utilize the other sugars, we don't have any industrial strains that are able to utilize all the sugars to make ethanol at an acceptable level. So this rather complex pathway illustrates the fact that it is possible to imagine integration of glucose and xylose fermentation to ethanol. And now much of the work in this field is associated with understanding the regulation of these and several other related pathways and understanding how to integrate them into an integrated pathway for sugar fermentation of all sugars into industrial microorganisms. And I think that these are the kind of problems that are probably just reflect the fact that not enough work has been done. I think no miracles are going to be required to understand how to integrate these pathways. And this is where recent advances in synthetic biology and systems biology are likely to be very important for making progress. Now, to return to this scheme, what I talked about so far was several elements, aspects of the process, such as altering the amount of energy that has to go into breaking down the biomass and utilizing all the sugars. There are other places in the overall process where improvements are possible. And probably the big one, or one I'd like to talk about next, is the question about whether ethanol is even a desirable end product. I would say that although ethanol is the big biofuel today, that's primarily a historical accident, that we had a kind of a brewing and alcoholic industry that was able to use sugar to make ethanol with pretty high efficiency. But in fact, at this moment in the development of the biofuels industry, it's good to step back and say, what is it that we would really like as a fuel? Ethanol has a number of problems. For example, if you get to ethanol in this process, it's usually about 12% ethanol and 88% water. And to turn that into fuel, you have to distill the ethanol. So, of course, distilling requires a lot of input of heat, and that decreases the overall efficiency. So, from first principles, what you would like is something that's a lot more hydrophobic than ethanol, so that, for example, it will partition out of the aqueous phase spontaneously. That would also create improvements in the overall fermentation process, because unlike ethanol, which is toxic to the cells, it's possible to imagine having cells produce hydrophobic compounds that partition out of the aqueous phase and are therefore not toxic to the culture, and the culture could then be presumably maintained in a continuous culture for a long period of time, and the amount of energy that goes into creating the cellular biomass of the culture would be decreased. Additionally, ethanol is not desirable because it's miscible with water, and therefore it corrodes pipelines or containers. And indeed, in the United States, we have about 240 million cars or vehicles of various types, and only about 5 million of those can use more than about 10% ethanol in their fuel because of the miscibility issues. If we're ever going to move towards the Secretary of Energy's goals of having 30% of our fuels from biofuels, if that were ethanol, if that fuel is ethanol, then we're going to have to ultimately replace most of the fleet, which would be a huge infrastructure cost. The fleet turns over rather slowly. I think the half-life is 7 or 8 years. So from first principles then, it would be desirable to make something that's a lot more hydrophobic than ethanol. Now nature offers many such alternatives. For example, as shown here, plants, algae, and bacteria all synthesize compounds such as alkanes and long-chain alcohols and waxes that are very hydrophobic. Indeed, the principal component of our current fuels are alkanes. I think I've already mentioned the second point on this slide, that if we could produce such hydrophobic compounds, that would alleviate the energy required for dehydration and could reduce toxicity. So here's an example of how organisms make alkanes. On the top is shown a typical fatty acid. All living organisms make fatty acids because the membrane lipids are composed of long-chain fatty acids. It's known that many organisms have a class of enzymes called decarbonylases that are able to remove the carboxylic acid function in a two-step process to produce an alkane, as shown in the bottom. For example, the surface of all plants is covered with alkanes as part of the wax that prevents plants from evaporating. Similarly, many microorganisms make such things. And however, interestingly, the identification of the decarbonylase has never been accomplished, mostly because really very few people have tried since the mid-1970s when these enzymes were discovered as an activity in crude extracts. And I think it's a perfect example of an opportunity, a scientific opportunity that could fundamentally change the whole bioenergy spectrum that has emerged, or re-emerged, I should say, as an interesting problem, something that's just been sitting unexplored basically for the last 25 years. So if I were to summarize what I tried to convey up to this point, I would say that in order to make biofuels, cellulosic biofuels feasible, no miracles are required. There's about four main things that need to be done. One is, it would be desirable in the long run to modify plant composition to minimize the energy required for depolymerization. And I tried to give you the example of, well, maybe we could modify the structure of lignin so that we could remove it under physiological conditions. We need to identify better catalysts for breaking down the polysaccharides. And I would say that that's an area also that's been very poorly explored. There has been quite a bit of work on a small number of enzymes for very mechanistic reasons, but we don't really know yet how much diversity exists in nature. And so now there's an opportunity using the high throughput methodologies to go out and explore what's possible, or what nature produces, and bring them back to the process. I think we need to develop new industrial microorganisms that can ferment all the sugars, and also organisms that can be resistant to certain inhibitors that arise during the degradation of biomass, and can thrive in large-scale industrial reactors. And finally, I think there's a big opportunity to develop microorganisms that can secrete what I'm going to call second-generation fuels, such as alkanes or terpenes or various other hydrophobic compounds that will not require replacement of the current infrastructure. I think the field has many questions, although the general field of biofuels has been around for some decades. It's been very poorly populated. There haven't been a very large number of people working in the field. The price of oil was too low to really encourage societal support of research in these areas. So, so many questions remain. I think one question that comes up is how challenging are the technical problems that I've alluded to? And there's no short answer to that until the problems are really solved. But based on my experience as a scientist, I personally believe that we will be able to make a price competitive biofuel process probably within the next 5 years. And I think we will be able to make biofuels actually much less expensively than we currently can make fossil fuels, at least from petroleum, within 20 years. I don't see any immense technical barriers. That also addressed the question of what the time frame is. I think we'll have plants in 5 years. I think we'll have big plants and quite widespread cellulosic biofuels within 10 years. It's frequently also asked, well, you know, why is there a focus on biofuels as opposed to other things such as conservation or wind or photovoltaics? And I want to make it clear that I'm not proposing that biofuels is an alternative to any of those things. My own experience and perspective on our energy needs is that we need all possible carbon neutral technologies. We need to put them on the shelf so that they're available to us. And biofuels are of particular importance in the context of transportation fuels, where it's very challenging to, for example, use electricity, which can be generated from wind or nuclear or other sources, as a transportation energy source. So the remaining type of question that comes up is the degree to which there are risks. And from the technical side, I don't see any particular risks associated with this. I think that the kind of work that needs to be done is typical industrial microbiology and protein engineering and enzymology. So I see this as a kind of a technology that can go forward within the existing structures and shouldn't create any unusual societal concerns. Well, I hope that gave you some sense of what's going to be required to make biofuels a part of our energy economy. It's a very interesting topic because it's so multidisciplinary. There's elements of chemistry and genetics and agronomy and microbiology and socioeconomics. I find it to be one of the most interdisciplinary kinds of areas of endeavor that I've experienced in my career as a scientist. And I think it's in some ways a very interesting insight into perhaps how going forward the new technologies of the future are going to be very multidisciplinary. Thank you very much. I hope that was informative.